Hello, everyone, and welcome to today's live broadcast, Hematopoietic Cell Therapy Products, Determining Potency and Stability. I'm Christy Jewell of LabRoots, and I will be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by STEMCELL Technologies. For more information on our sponsor, please visit stemcell.com. I would like to remind everyone that today's event is interactive, and we encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. Also, please notice you can share this webinar on your personal social media. Just click on the Social Sharing tab on the left to let your friends and colleagues know about today's live event. Now, if you have any trouble seeing or hearing the presentation, click on that Support tab down at the top of your presentation window, or you can use that Ask a Question box to let us know you're experiencing a problem. This presentation is educational, and it does offer continuing education credits. Please click on the Continuing Education Credits tab located at the top right of your presentation window and follow the process to obtain your credits. I would now like to introduce today's speaker, Dr. Jackie Damen, Senior Scientific Advisor, Hematopoiesis, STEMCELL Technologies. For a complete biography on Dr. Damen, please visit the Biography tab at the top of your screen. Welcome, Dr. Damen. You may now begin your presentation.
Thanks, Christy, for that introduction. Let's dive in now to a STEMCELL-sponsored webinar today, Hematopoietic Cell Therapy Products, Determining Potency and Stability. The learning objectives for the webinar today will really be to define the CFU assay and its characteristics as a biological tool in identifying hematopoietic stem and progenitor cells, to recognize the utility of the CFU assay in processing and manufacturing of hematopoietic cell therapy products, and also to identify publications using the CFU assay to verify the potency and stability of products. Just to give you an outline of the sections we'll go through today, we'll start with hematopoietic stem cell therapy and do a bit of a review of the regulatory requirements in this space, then have a really short review on the applications of expansion of hematopoietic stem and progenitor cells ex vivo. We'll then really dive into the assays to quantify hematopoietic stem and progenitor cells. In more particular, in section 4, discuss the CFU assay, applications of the CFU assay, and especially applications when it comes to potency and stability. And then we'll end with a little bit of tips and tricks on how to standardize the CFU assay. Okay, let's start then with hematopoietic stem cell therapy. So as we know, there's a long history of bone marrow transplants in the treatment of hematological disorders. This is a long history that started really in the 50s with the first bone marrow transplant to cure leukemia. And we know that it's been a curative treatment for 40 years, more than 40 years, for blood cancers such as leukemia and lymphoma, bone marrow disorders such as aplastic anemia, as well as genetic diseases like sickle cell anemia. In fact, bone marrow was the sole source for many years, and as you can see by the graph here on the right-hand side, which shows the number of transplants in the U.S. from 1992 to 2016, the blue bars indicate bone marrow as the source. And up until 2000, it was the only source, but in 2004, mobilized peripheral blood actually became more of the major source for hematopoietic stem and progenitor cells, really due to advances in technology that has allowed the ability to mobilize and collect large numbers of hematopoietic stem and progenitor cells in a less invasive way. Cord blood has also popped up here, and you can see that by the orange bars, particularly around, again, 2004, really from evidence in 1996 to 2001 that cord blood and bone marrow actually they showed that there was no difference in leukemia-free survival rates when looking at treatment for both children and adults, recognizing in most cases this was young adults, because the limitations, of course, with cord blood is that you can only collect so much blood and only get so many cells. When it comes to some stem cell therapy products, one has to recognize that there are requirements for these products. When it was fresh and it was bone marrow, these requirements were not historically required because everything was done fresh. But now that things like mobilized peripheral blood and cord blood have entered this arena, we have to recognize that these cells are somewhat processed. Red blood cells are often removed, and in some cases, these cells can be manipulated and changed with the new therapies. So we really have to recognize that you have to really look at stem cell-derived cellular products and ensure that there's reproducibility and consistency, which is inherently a problem due to the donor variability of the starting material. So it's important that the product quality, both the purity and the potency, needs to be established at various stages of manufacturing. And the purity means really the cell composition, including the contaminating cell types. And these are typically identified using flow cytometry. The potency needs really to identify the capacity of the product to affect a given result. And the FDA generally doesn't accept viability as the only measure of potency because there's issues with assuming that viability means that the cells can function. If you look here on the right-hand side at the table, this is really a outline of a table that examines the release criteria after retrovirally transduced hematopoietic stem cell product. And this is from a recent paper from Molecular Therapy in 2017. And you can see that the requirements for safety testing include sterility, of course, purity, identity, and potency. And for purity, they use flow based on really the only marker that identifies hematopoietic stem cells, and that's the CD34 positivity. And for them, they wanted their release criteria to be greater than 90% if possible, or report the value, recognizing that when you do the clinical trial, you'll actually find out if 90% is actually necessary, or if you can get away with something like 85%. Usually doing the clinical trial allows you to really identify the ranges that are acceptable for the purity of something like this. With regards to the potency, the viability has been identified as being required, and in this case, greater than 70% post-thaw, very specifically using trypan blue. But they also ask that the CFU assay be done and a value reported. But really, what value would that be? And the fact of the matter is, it really kind of depends, again, on the treatment that you're doing and the manipulation that you're doing. So if you started with cells that had a high CFU frequency, then post-modification, you would really want to expect that reporting the value that there would be a percentage that would be acceptable, like 80% or something. So these are really key, important measurements that need to be confirmed before you can release products for therapy. In fact, if you look at the guidance for the industry potency tests for cellular and gene therapy products that the FDA has released, one very important statement in 21 CFR 11 is that no lot of any licensed product shall be released by the manufacturer prior to the completion of tests for conformity with standards applicable for such product. These mean, again, and you can see it's the same as the last slide, that these tests include values for potency, sterility, purity, and product identity. And really, if you look at what a potency assay should be, a good potency assay, it should be something that you can actually validate. It should also demonstrate product activity. It should have a quantitative readout, and it should also indicate product stability and consistency. Normally, these start, as you can see, at the beginning of the discovery phase when you're developing a therapy. Potency assays are really at the developmental stage there. And then as you go through your clinical trials, the more assays that you have that describe the potency of the product, the more able you are to find one or two that actually very specifically correlate with good clinical outcome. So when we look at, for example, one of the only, at this point, FDA-registered cell therapy products, these are cord blood cells, we recognize that there are very strict regulatory requirements when using cord blood products for basically hematopoietic cell therapy. All cord blood banks, for example, need to be FDA registered, and most are also accredited by AABB, in fact, other regulatory bodies. And both of these require that you test cells pre-freeze as well as post-thaw to really ensure that the potency of the cells are good, and they recommend using the CFU assay, as you can see in these guidances that are displayed on this slide. So let's then talk a little bit about the expansion of hematopoietic stem cells ex vivo and why this is so important to cell therapy products. Okay, really breaking it down to the therapeutic applications of the expansion of hematopoietic stem cells ex vivo are listed here. And the reality is expansion of hematopoietic CD34 cells have really been the holy grail of hematopoietic research for many, many years, and mostly because of the important therapeutic ramifications. So expansion of CD34 means the expansion of the long-term hematopoietic self-renewing stem cell that can really repopulate, as you can see by the diagram, all the blood cells in a compromised host. So expansion of CD34 from cord blood has actually been looked at for the last 20 years to really try and improve, since cord blood has a limited number, to improve the numbers of hematopoietic cells that have that engrafting function, as well as improve the recovery time for allogeneic transplants in adults with leukemia, and really open up that pool of cells that could be used to treat those patients. Also, of course, what we've learned from how to expand cord blood can be transferred to how to expand mobilized peripheral blood, and that's going to become important as people try and scale up gene therapy applications for autologous transplant options. And then there's also been a lot of therapeutic applications for hematopoietic stem and progenitor cells when it comes to lineage-specific differentiation, and a lot of research that really started in the 80s and 90s, both focused on the conditions required to expand hematopoietic cells in culture so we could really study them. That led us to understand a lot of the conditions required not only to expand cord blood and other CD34 purified cells, but also led us to understand what are the critical factors to really induce lineage-specific differentiation. This is important, and there's been always this clinical application of doing clinical scale ex vivo production, for example, of red blood cells and platelets to have a safe and adequate supply of blood cells. We know all the conditions. The issue and the reason it's still being studied today is really how do we optimize this and make it cost effective. The other one, of course, that is booming right now and is probably really only in its infancy is the generation of lineage-committed genetically altered cells for targeted immunotherapies. So we're actually also now looking at using hematopoietic stem cells and properties of T cells, for example, to really increase the quantity of those cells genetically and genetically engineer them so they can be targeted not only for blood cancers, but other types of cancers. So a lot of applications based on just the expansion of hematopoietic stem cells ex vivo, and due to 30, 40 years of research, we now have a lot of knowledge about the conditions that's required. In fact, one thing that is a certainty when it comes to, that we've realized that when it comes to trying to expand hematopoietic stem and progenitor cells ex vivo is being aware of the regulation of hematopoietic stem cell self-renewal. So whenever you put these really rare hematopoietic CD34 positive cells ex vivo in culture, the certainty that we've realized over the last 40 years is those cells start to lose their stemness. And it's really because of the fact they've been removed from the niche. And there's a lot of niche factors in the niche that we're still finding, actually, which ones are critical. But most of them involve either intrinsic factors like transcription factors or extrinsic factors like growth factors, cytokines. And what we've realized more lately are small molecules based on the fact that we've now identified that it's really the self-renewing population and that first symmetric division, as you can see in the diagram on the slide, that is really critical to allowing the very specific hematopoietic stem cell to divide and maintain its ability to engraft in a host. Because if that cell has an asymmetric division, then you start that process of differentiation and lineage maturation. So the key is, as soon as you put hematopoietic stem cells in culture, they start to differentiate. And really, recent evidence in the last 10, 15 years has allowed researchers who have been studying this phenomenon for many years to identify novel small molecules, which we call differentiation blockers now, that have allowed one to really find factors that will allow that first self-renewing division and expand that population more than we've ever had been able to in the past. So to be able to show that, of course, a lot of the research has depended on a lot of assays of how to quantify hematopoietic stem and progenitor cells. So I'll just review those, as well as outline some of the applications of how those can be used. So if we look at this slide, we realize that there really are two basic ways to quantify hematopoietic stem cells. The first is phenotypically, and that really measures physical cell properties based on cell surface markers that those cells have. And as we've mentioned, expression of CD34 is really the most common marker used to identify hematopoietic stem and progenitor cells. And as cells differentiate, they lose that expression of CD34, but they also then become positive for other lineage markers that are specific more for the mature cells. And a lot of these are listed here, and you can see based on the images which of the cell types and which of the lineages express which of these, what we call lineage markers. So to be able to identify true hematopoietic stem cells, they should be CD34 positive and lineage negative. And we've since learned that there are other markers that we can look at, like CD90 positivity and CD49F and c-KIT, that will really help define different subsets of the CD34 population that have some of which we really want to define are those that are in the engraftable population versus those that are just starting to become fate committed. Better than a phenotypic assay, especially when you're trying to prove something, is a functional assay. So really there are a couple of properties of hematopoietic and stem and progenitor cells that really define those populations that allow us to measure them based on those functions. And again, thanks to 40 years of research that has showed us what those are. And for hematopoietic stem cells, that's really an in vivo transplantability or engraftment assay, because that's our expectation, that the hematopoietic stem cell can engraft a compromised host. So there's models now to show that and measure that, as well as in vitro assays, which are a little easier to perform, but allow us to really identify progenitor and stem cells based on their very unique ability to still have the ability as an adult stem cell to proliferate and differentiate into all the blood cell types and all the cell blood lineages. So in this slide, you can see that there really is two main assays that are used, and pretty much all publications these days actually show both of these if they are looking for effects on hematopoietic stem and progenitor cells. The first actually is called the SCID repopulating assay, or which is an assay that was really developed in 1997 by John Dick's lab, and it really is an in vivo quantitative assay that measures the engraftment potential of the hematopoietic stem cell to repopulate the hematopoietic system in a compromised immune-deficient xenotransplant mouse model. So it's named after the original strain that was used, which was NOD-SCID. Today, however, there's been a lot of improvement on that strain, and you can get all kinds of versions of what we call NSG mice now that work in the same way. The other is the colony-forming unit cell assay, or the CFU assay. That really measures the progeny of that long-term hematopoietic stem cell. So the SCID assay really measures just that ability of that long-term hematopoietic stem cell to repopulate a host for the lifetime of the animal. But the colony-forming assay really looks at the progeny of that cell, which are the progenitors that actually control and maintain the right number of blood cells on an everyday basis. And then this assay is actually a clonal assay, which then means by default or definition that it is a quantitative assay that identifies the frequency and lineage commitment potential of functionally viable hematopoietic stem and progenitor cells. And I've just included some references here so that you can actually take a look at how long ago these were done and how impressive that is. Okay. So when you look at assays to quantify hematopoietic stem and progenitor cells, really the workhorse has been the CFU assay. It demonstrates hematopoietic stem and progenitor cell activity. It's a clonal assay. When you look at the diagram here, you'll actually see some images of measuring a progenitor cell. So the difference between a progenitor cell and all the mature cells is that unique ability to grow. If it's a very early progenitor, it might have multi-potential, which means that it actually can make cells of different mature cells of different cell types. And you can see that by the CFU GEMM image shown here, where you see red cells as well as other cells surrounding it, versus the ability to actually look at more lineage-specific progenitors. For example, BFUEs are from the erythroid lineage and CFU-GM from the myeloid lineage. And the ability to actually watch a single cell in semi-solid media expand and grow in culture in a spatially identifiable and separated way then allows you to really count and look at what is the frequency per cells plated, and am I really getting the appropriate and the normal erythroid and myeloid ratio, as well as platelets. You can also look at progenitors for CFU-MK to confirm that not only the cells are healthy and working, but if you've manipulated them, has any of that changed? So it's a clonal assay which makes it quantifiable, and it gives you the frequency of erythroid, myeloid, and platelet progenitors, megakaryocytes, in really the ratio of, if you have a progenitor, it's one colony and it's one CFU. So that allows you that measurement. And through the years, of course, this assay has been used in many different ways. And if you look at the references that we've included on this slide, you can see that in some cases, people that have looked and compared what they see in a CFU assay to the outcome in patients following bone marrow transplant, they can show that the CFU assay is a very good potency assay that strongly correlates with the engraftment potential of these hematopoietic stem cell products in patients. When it comes to applications for quantifying hematopoietic stem and progenitor cells, this is actually something that people that are looking at novel therapies, especially using gene therapy, have used to really test the efficacy of their manufactured product. So really, if you're looking at purified, if you're wanting to treat patients, for example, with a genetic disorder like sickle cell anemia, you can purify the CD34 cells from the patient, and then with genetic editing, basically change those cells, correct them, in other words, manipulate them, but then they also need to be expanded before they're returned to the patient and potentially cure that patient's disease. How much expression do you need to cure that disease? Of course, depends on the disease. So assays really then can allow the ability to give indicators of a successful transplant prior to engraftment in vivo by using the two assays I just described. The engraftment assay, which can assess the long-term effects of the gene therapy that you've done. You would want to make sure in a mouse model that the expression of whatever it is that you've changed actually is long-term and will be expressed for the lifetime of the individual, but also how much of it needs to be expressed to basically make a change so that it cures the disease. And this can all be confirmed using that assay to confirm expansion of cells that still have that engraftment potential. The CFU assay is also important and easier to do, of course, and it will allow the assessment of the progenitor frequency post-manipulation and make sure that you get recovery and normal recovery and function of all the functional ability of those cells to differentiate to all the different mature cell lineages. So when it comes to the applications, actually, of these assays that quantify hematopoietic stem progenitor cells, I really encourage you to look at this publication as shown on this slide. It's really a good case study of identifying the challenges that one needs to think about when they're getting into the gene therapy space and trying to make cells, hematopoietic stem cell products to help cure people with these genetic disorders. And this publication really highlights the large-scale production challenges that exist when trying to genetically engineer hematopoietic stem cells, really recognizing that one of the things you have to overcome is a limitation to the number of cells that you have. Typically, patients with disease don't mobilize very well, so it's important to know the conditions and optimize those to get enough cells to allow complex genetic manipulations without losing the stem cell function. So this paper really defines the optimal clinical and clinically applicable strategy to purify hematopoietic stem cells and determine conditions for ex vivo expansion to preserve that important engraftment function. And I really encourage you to look at this paper, because it also shows that when you're trying to expand cord blood or bone marrow CD34 cells, there's some pretty dramatic differences. Cord blood has a lot more proliferative potential, so it can be expanded, you get more cells, and you can do it for longer. You have to be really careful if bone marrow or mobilized peripheral blood is going to be your source, because those don't expand as well. You have to really understand you have to use these sort of small molecule inhibitors that have recently been identified that are promoters of hematopoietic stem cell self-renewing divisions to really get enough cells for that. So I've summarized that actually on this next slide. This is a busy slide, but I really wanted to highlight here that you really have to look, and this paper defines it very well, the protocols and the tools and how to standardize those for your expansion application to make sure that it's efficacious for gene therapy. Some of the protocol things you have to think about, for example, is to standardize your workflow. What are the subtypes of CD34 are you going to purify? Are you just looking at purifying total CD34 and modifying all of these? And then optimizing the expansion time. As I mentioned, they really highlight using the engraftment assay than the NSG mouse assay, that less is better when you're trying to actually maintain the potency of that engraftable hematopoietic stem cell. Included are, of course, optimizing your treatment protocols. What cytokines do you add? What is the cocktail you want to do? Are you going to add small molecules? How much expansion do you need? And in this publication, they actually highlight that the small molecules, SR1 especially, has allowed a huge amount of expansion that they weren't otherwise able to get without adding these molecules. And also recognizing that it enhanced transduction efficiency for the viral vectors that they used. So thinking about these things and optimizing your protocols and strategies for expression of the genetically modified population is really important, whether you're working with viruses or doing non-viral approaches. The tools and reagents are obviously also highly important, making sure that you have a consistent supply of optimized media. For the case of expanding CD34 cells, these should be serum-free culture medias. And also looking at the early acting cytokines. Which ones are you going to use? And in fact, about, again, 30, 40 years of literature of labs looking at what early acting cytokines to use. This has not changed, honestly, since the 90s. Everyone at that point identified the three main players of stem cell factor, FLT3 ligand, and TPO. Some have added IL-3, some have added IL-6. What's changed in the last 10 years is, in most cases just in the last 5, is this idea of small molecules. In other words, identifying and screening for molecules that we know are perpetuating that self-renewing population is still something that, and the reason why hematopoietic stem cell expansion is still the holy grail in research. We still don't understand the mechanisms of these, but we can certainly tell that they are important in the expansion, and you can really see an increase in the number of CD34 cells you get using these so that you have a lot more cells to do manipulation. And of course, once you do your manipulation, making sure that you have good assays that are going to tell you if it's working. And I've outlined those for you using the skid repopulating cell assay for engraftment, as well as understanding the purity, making sure you have enough CD34 cells as identified by flow or any other contaminating cell populations, and also looking at the ability and functional ability using the CFU assay can really identify that you have maintained a good frequency of those progenitors and you haven't actually changed or lost any of the progenitor types. So you've heard a lot now about the CFU assay. I'm going to dive in a little bit now and talk more specifically about the CFU assay and describe its some publications that show the usefulness of this assay, especially when it comes to looking at potency and stability. So the CFU assay has a lot of applications, and if you remember at the beginning of this talk, we talked about the fact that the FDA has very specific guidelines for cord blood banks that are making hematopoietic cell therapy products. And there are actually, to date, a number of cord blood cell therapy products that have been approved by the FDA. You can see an example here of an insert from one of these, packaged insert from Alicord, which is a supplier of one of these approved cell therapy products. And that insert and inserts for all of the products that cell therapy or cord blood cell therapy products that I've seen actually include information and very detailed information on how to receive the product, how to prepare the product, how to dilute the product, as well as how to infuse it and ensure that you actually maintain a sample that you can do QC on so that you can confirm that the product that you have has not been affected from the transport from the cord blood lab to the clinical site. This is really important that you put in temperature monitors when monitoring systems when you're shipping cells, and it's actually up to the cord blood bank, as you noticed in the other guidances, to ensure that they've looked pre and post-thaw, that they have hematopoietic stem and progenitor cells that are actually going to grow out, and they use the CFU assay or analogous potency assay, but they recommend the CFU to ensure that that's the case. They want to confirm that, though, that if they've shipped it to you, typically that happens in cryo shippers, that if the temperature monitor says it's okay, that is probably a good indication that the product that you have in your hands is good. But assuming that you don't thaw it inappropriately and that it's thawed well, that this assay then can really tell you at the end what the product that the patient got was and what was the stability and what was the potency of that product. So when you're looking at the quality control here, it asks you in the product insert to make sure you do a nucleated cell count, definitely a viability test. You want to make sure that the cells that you're injecting are viable. In fact, when you order these, they kind of give you the idea of, well, this unit is the right one that you have, the right compatibility for your patient, and it has X amount of cells. And that's important because you know the weight of your patient, and it's one of the ways when you're doing a transplant to ensure the patient gets the right dose. It's really based on the viability, and more particularly the viability of CD34 cells. So those have to be part of the quality control test. They also ask you to do the CFU assay, of course, for potency, and a microbial test to make sure that you have the sterility. So these QC tests really are because the product is cryopreserved, and you want to make sure that it is stable, that it's transported okay, and that the product that you gave the patient is actually, you can confirm with the CFU assay, was actually a good product. In fact, cord blood banks now, and a lot of people and a lot of institutes that are freezing and using and storing cell therapy products are now have to write into their programs that you need some kind of stability test that is actually performed annually to ensure a good quality product. And those are new regulations from JC that came out in 2015. And this slide actually shows you that the CFU assay not only is important as a QC test, but as I mentioned, the dose that's used when you're actually collecting hematopoietic stem cell cells before you're going to use them for therapy, you usually base that dose on the number of CD34 cells. And historically, that was done, of course, because we were talking about bone marrow that's fresh. Or in the early days of mobilized peripheral blood, that you could actually do flow, identify the purity of CD34, do it right at the time. It only takes an hour or two to do flow. And that way you can ensure that if you needed to mobilize the patient for longer and that their CD34 wasn't up to the levels that you needed them, you could actually use flow more specifically to make sure that you did get enough cells from that patient, if at all possible. In some cases, patients that don't mobilize well are actually then recognized after 3 or 4 days of mobilization with GCSF that it's not enough. So they're actually asked the patients get Mosabil to really improve the number of CD34 cells they have. So for transplantation purposes, they try and get a dose of at least 10 to the 6 CD34 cells per kilogram. And that is, not everybody can measure the CD34 cells the same way. Flow can be a little bit unstandardized, and different people can look at it differently based on gating strategies. So there was a protocol that was actually published in 1996 to try and standardize this. Unfortunately, surveys have showed that not everybody uses that protocol. But nonetheless, it actually, when you look at a lot of the clinical trials and successful transplants, it usually sort of recognizes that if you inject 10 to the 6 CD34 cells, that should be a good dose for the transplant. But injecting it is one thing. Waiting to see the success of the transplant is another. You won't actually see the success of a transplant until about 2 to 3 or 4 weeks after the patient has been injected. And at that point, it's good that if you've set up the CFU assay at the same time, you actually have confidence as to whether it was the cells that was the problem or everything else that the patient has to go through in that transplant process. So the CFU assay has always really provided that data. It's retrospective, yes, information about the functional activity, but it's something that's quite critical when you think you've had the right dose, you've injected your patient, and then you realize 3 or 4 weeks later they're not engrafting. If you have set up the CFU assay and you had no growth, you'd be very quickly looking for another donor. In fact, some recent publications by Dr. Watts and Lynch here, and I show this, this reference is included down here, that if you've got a poor mobilizer, and this happens quite a bit with patients with disease, you basically have no choice. You inject them anyway, and if you've done a CFU assay, typically what you find that correlates with good engraftment is when patients actually can present with 2 by 10 to the 5 CFU GM per kilogram. So if you go back, look at the cell numbers and identify, yes, we have greater than 2 by 10 to the 5 CFU GM that we injected, then you know that maybe the engraftment is just taking a little bit longer and you're not looking for another donor right away. So it does give you that retrospective data, but it's a lot of comfortability to make sure the assay did work, that the transplant, at least the patient got the right cells in that case. The other, of course, issue is that even more recently, we've realized that CD34, when you're looking at viability on cryopreserved samples, works until it doesn't. And this slide really illustrates a situation where it didn't. And there was some issues from a hospital, the Great Ormond Hospital in London, where they had realized that there were patients with delayed engraftment who had received adequate post-thaw viable cell counts, but they couldn't understand what was going on and why their transplants were delayed. So there was actually an investigation that was started because of this, and this sort of outlines sort of what happened as far as the BBC news that came out on this incident. And Dr. Mike Watts, again, was involved in investigating this and really did an independent investigation and a publication to show that the assessment of post-thaw CD34 viability is unlikely actually to be adequate to really guarantee functional activity. And he recognized, and you can see that in the diagram here, based on freezer profiles, that if he took cells and froze them at different centers and by passive freezing methods, compared to the freezer program that they were using at the Gosh Hospital, where these patient cells had been collected and stored, that you could see that there was definitely a difference in the freezer program profiles, that when you look at the Gosh Hospital, the temperature dropped very quickly, and then in fact, in a non-optimal way, you really want, when you're doing freezing programs for hematopoietic stem cells, to really freeze them at a degree per minute, and that has to be maintained till at least past minus 50 degrees, otherwise that can really affect the functional output of the cells. The CD34 viability looked great, but when he actually compared the CFU-GM or CFU output from these different freezing profiles, recognized that the data showed really that all of them were fine except the one from the Gosh rate-controlled freezer, where that steep curve in the freezer profile was observed. There was actually no growth of the CFU in those samples, whereas the CD34 actually showed some decent viability. So it's one of those things that, yes, CD34 viability works until it doesn't, and if you really want to be sure on thawed cells that you've got cells that are able to grow like they would be expected to do in the transplant in the patient, then you really have to perform some other kind of potency assay. In that review, Mike Watts also reviewed some of the literature. I've shown a couple of references here, where if you look at people that have really investigated the effects of storage conditions on hematopoietic stem cell potency, you can see that they all come to the same conclusion, that the assessment of the viability of CD34 frequency can fail in some cases to predict the effects on potency, which can be observed using the CFU. So a lot of the papers will always validate whatever process they're doing based on the CFU assay. For example, a paper from Yang in 2011 concluded that if there was a problem with the rate-controlled freezer and there was any interruption, you would definitely have to use a CFU assay to affect the potency and the effects on potency of the product, because CD34 failed to predict that. And also in a paper from Jansen in 2009, he investigated the transport of cells, because in a lot of cases, apheresis is not necessarily done in the same place where the patient's bone marrow transplant is performed. So these cells tend to move around, and it takes a little bit of time to move from where they're collected to where they're possibly going to be stored and or frozen. And he evaluated, and you can see in the orange box in the table on the right, that if you stored these cells for 48 hours in anything warmer really than 4 degrees, there was an effect. In fact, if you look over 10 degrees, you can see that viable CD34 was still 80% on these fresh cells, actually, but the CFU-GM output was only 50%. So it really then goes to show you that if you're actually looking at different processing and different storage conditions, and you expect samples to actually not come to you immediately, that you really have to think about using the CFU assay to validate all the processes you're doing, not only in the manipulating of the cell stage, but also after the manipulation, between getting those cells from where you're manipulating them to where you're actually putting them in the patient. And just to summarize, really, this slide shows that the CFU assay then is a good potency and stability assay, because it does address all these factors that affect the potency and stability of hematopoietic stem and progenitor cells. And that is the mode, the time, and the temperature of collection and transport, the storage conditions, the processing methods, the cryopreservation protocols. These are critical. So if you want to actually qualify your rate-controlled freezer, again, you have to use the CFU assay to get an appropriate response on how that affects the quality of the cells, as well as your protocols, which will depend also on the number, the volume, all kinds of things. So just to summarize then, the CFU assay has been used to identify the potency and the quality of hematopoietic stem cells. It's correlated very well with engraftment, and for years, researchers actually use this assay to evaluate all kinds of factors and biochemical pathways involved in hematopoietic stem and progenitor cell engraftment, mobilization, and homing, and really evaluate genes that were critical to the maintenance of hematopoietic stem cell homeostasis depended on knockout mice, and recognizing that in those mice, when genes were knocked out, what effect did that have on the output of the progenitors? Well, usually the CFU assay was the readout. In addition, the CFU assay, because it's clonal, allows you to really look at changes in the frequency and the morphology of hematopoietic stem cells on a clonal level. And for hematological diseases, for example, this has been an advantage. For something like polycythemia vera, which has a JAK mutation, it's not in every cell in the body, but to identify the percentage and get an idea of what we would call disease load, you can actually pick colonies after the CFU assay and identify and get genetic material from those and use that for PCR to really evaluate mutations or do any other kind of genetic analysis. Another sort of use of the CFU assay, especially when you're looking at gene therapy approaches, is to recognize that you can identify the frequency of your particular manipulation, and is that high enough for those cells and for that disease to be corrected in general. So a lot of different aspects of the CFU assay can be used, and one of the ones also shown in the diagram here is the ability to evaluate the effects of toxic compounds. And you can see, for example, in this example, that they looked at the expression of the mutation and could identify that in BFUEs or erythroid colonies that had the mutation, that when you added the drug, those were really eliminated, where there was very minor effects on samples that didn't have that mutation. So just before we close off then, I just wanted to give you some tips and tricks about the standardization of the CFU assay. As we recognize from this presentation, it's definitely a very versatile assay, can give you a lot of information. It hasn't been used more widely because of the sources of variability in setting up this assay. And originally it was more about media formulations. In the old days, we didn't even have recombinant cytokines. So making sure that you're always using a media that's qualified appropriately. Methicellulose has been the go-to for the last 25 years. And STEMCELL Technologies, of course, has been offering qualified methyl media for 25 years. And we qualify ours using primary cells, so the cells that you are actually using for these therapy products. You also have to make sure you have standardized practices, ensuring that your staff is trained on all the procedures. And some procedures that you really have to think about are, how are the cells received? How are they processed? How are they thawed? What is your, what are you using for your viability stain, for example, as well as how many cells to plate for the CFU assay. It is linear in certain ranges, so it's always good to set up a couple of different concentrations to make sure you're in that right linear range, but it is quantifiable, so it can be validated. Include when your SOPs for something like this, to make sure that it's done the same way all the time, the cell source that you typically use. What are your expected viable ranges of the cell and cell numbers? What's the appropriate media formulation that you want to put those cells in? And we make this easy by offering optimized formulations depending on the cell source. And of course, what are your ranges for plating concentrations when you set up this assay? As I mentioned, it's linear, but only in certain ranges. So just to give you an example, I've outlined some ranges here, but you can see for CD34 enriched cells, you need very, very little cells in this assay. So typically you're setting up a lot of assays to characterize those cells. The fact of the matter is you're always going to have enough for the CFU. You don't need more than 500 per dish, honestly. And if you can set that up pre and post engraftment, that's going to give you a lot of information about what changed during that modification. One of the other variables when it comes to the CFU assay is scoring. So you can see by the images on this slide, this gives you an example of a plate of cord blood mononuclear cells with about 80 colonies. And scoring these is not simple, so you don't need any fancy equipment. You just need an inverted microscope. But what's more important is that your staff is well trained in performing the assay and identifying the colonies. And really that also means typically if you're doing this for clinical reasons, that they participate in regular training such as proficiency testing, and STEMCELL. At STEMCELL we provide that as well. Also, we now have the ability to automate CFU colony scoring. That sort of eliminates that variability because it can really standardize and improve that reproducibility and repeatability as well as efficiency. But when you have different counters, different people sort of perceive colonies in different ways. When you use something like an automated scorer, especially with the algorithms that we've developed, that really does identify the colonies based on our knowledge of the colonies and how they look, but also recognizing how far away they are is really a mathematical calculation that is not easily done in your head. So it's really good to be able to visualize these, and with automated scoring you get this sort of image that has marked up colonies where the blue circles are the what we call multipotential GMM colonies, whereas the yellow ones are the myeloid and the erythroid are the red circles. So you can really evaluate that. If you would have scored it that way, you can review what the algorithm has done and sort of confirm that it's working for you. So just to give you a little bit more background information, the STEMVision machine was developed really in 2011 and has now been used by numerous cord blood banks globally. It accurately identifies and classifies and counts colonies from the CFU assay, from normal bone marrow, cord blood, and mobilized peripheral blood. And these cell sources have been validated for cell growth in methicult media, using more specifically the optimum formulation. More recently, in actually 2009, there was a cytotherapy paper that standardized, independently evaluated, and published. And you can see from the title here that validation of a semi-automatic device to standardize quantification of CFU colonies. So it really allowed us to say that it was more of a standardized assay, and you could use it. The automated counting would improve repeatability, interoperator variability, and comparison for colony counts compared to what you would otherwise get with manual counting. The other thing you notice from the images that it allows permanent archiving of some very high quality digital images of cultures so that you can keep these for all time. Anyone that's ever done colony counting recognize we do this on faith otherwise. And of course, it's always hard to take pictures of these because if you're not looking at the whole well, it doesn't give you the whole story. And we've also helped standardize this by synchronizing this with a lot of other products, especially our Methicult product. And just before I get off the CFU assay, I just kind of wanted to point out that with automated scoring, which really helps eliminate that variability you see from counter to counter, and what I've been seeing more often in teaching people to how to count colonies is that it's a situation where a lot of people that have been trained on this leave eventually. So to keep that consistency in the lab, it's nice to have something that does this automatically for you and gives you these images. As I mentioned, you get images that are actually, you first take images with the with STEMVision, and then after analysis, you get a sort of a data sheet that gives you the numbers as well as a marked up image that you can review. And every one of these has a sample ID qualifier for easy identification of what the sample was. And that really depends on what, of course, you put in. But once analyzed, you get a worksheet as well as you'll see this sort of Google, whatever they call it, image. But if you click on that, what it does tell you is once you've analyzed your images, there is a readout of what was the unit that you used, what was your plate ID, what was your barcode ID, if you use barcode, what was your user ID, as well as all the identifiers that you can put in are captured here and timestamped so that you know that these images were taken 14 days after the cultures were initiated, so that you're well within your QC parameters for setup and analysis. So I'm going a little over time, so I'm just going to summarize now and go back to the fact that can we ask the question and what are the answers? Does the CFU assay meet the requirements of a potency and stability assay? I'm going to start from the bottom because I think that's a little easier here. When you look at one of the criteria for a potency assay being that it should indicate product stability and consistency, I think that's a resounding yes for the CFU assay. As I've shown you, there's a lot of papers that whenever they're evaluating a new potency possibly assay, or they want to make sure that their whatever modification or storage condition or whatever they've done is actually working, the CFU assay is typically the assay that they use to validate their processes. And it's one that the regulatory bodies really ask when they go into the cord blood banks. How have you validated this process? Show me that you have actually used the CFU assay at least at some point to validate those processes, because it's such a powerful assay and it's been proven over many tens of years that it is correlates with the engraftment assay. The other thing a potency assay should have is a quantitative readout. The CFU assay, of course, is quantitative, it's clonal, and it does allow one to evaluate hematopoietic stem and progenitor numbers before as well as after transplantation. And if you can also use it to identify the effects of drugs, which I showed you briefly, or the effects of cytokines on cells. In fact, that's how we identified hematopoietic cytokines, was using the CFU assay, replacing conditioned media with recombinant cytokines to really identify their functional, what they do functionally. And if you compare patient samples pre and post manipulation or treatment, that can really get rid of that, as we discussed at the beginning, inherent donor variability for these products. Does it demonstrate product activity? Well, I think this is also an easy yes, because again, more than 30 years of research have shown that the CFU assay correlates with the engraftability. This was originally, of course, done in mouse models, but I've shown you evidence and publications that say, yes, this also is true when you move into translational studies and clinical trials. And can it be validated? Well, the CFU assay has been validated in some cases for different purposes, like the ability to identify maximum tolerated dose of chemotherapeutics by adding drugs into the CFU assay and looking at IC90 concentrations, for example. But in this case, it really depends on your application and the patient's response observed in the clinical trial. So how you're using the CFU assay, for example, if you've done genetic manipulation, you can first use it to understand you have a good product, all your cells are growing, but then you can go in possibly and pick clones and look at the number of clones that actually express the modification and identify a range possibly of which you would have observed from the clinical trials that is important in giving you a successful transplant. So it's always good to set it up at the front, because it could also not only be a potency and stability assay, but possibly also important in the identity of the product and possibly the release. So I'll stop now. Thank you very much for listening to this presentation, and I'll entertain questions.
Thank you, Dr. Damen, for your excellent and informative presentation. We will now start the live Q&A portion of our webinar. If you have a question you would like to ask, please do so now. Just click on that Ask a Question box located on the far left of your screen, and we will answer as many of your questions as we have time for. Wow, we have some great questions already coming in. Dr. Damen, let's start with this one. Can you define and give a small background about CFU assay?
Sure. Thanks, Christy. Yeah, that's a really good question. I didn't go into it a lot in this presentation. STEMCELL Technologies does offer training courses, and in fact, I'm usually the one that shows up for those. If you want one in your institute, of course, now we're looking at more virtual options, so hopefully those will be out soon. The reality of the CFU assay is, as a biological assay, is the concept of really being to identify those cells that have that proliferative potential ex vivo. So when you have mature blood cells, proliferation really slows down as those cells mature. So we know that, except with the exception of mast cells, mature cells don't really proliferate. So it's the ability to put those cells as a progenitor. So you would plate for bone marrow, for example, 10 to the 4 cells. That's a lot of cells, but only typically around 50 to 100 of those at that concentration are actual progenitors that will grow. So if you put that then in a semi-solid media where they grow in a spatially identifiable way, you can really see those as independent clones, as colonies. And if you look at the cells within those colonies, you can get an idea of the maturation. In other words, are these myeloid, are these erythroid, are these even granulocytes, or are these macrophages? Now, bear in mind, this is for the myelopoiesis side of hematopoiesis. There isn't really a good option when it comes to looking at colonies of the lymphoid lineages, and that's really because those cells do not completely mature in the bone marrow. They really are thrown out of the bone marrow as a like an immature pro cell. And in some cases, you can look at mouse pro-Bs, for example, in the CFU, but that's about it. But for human cells, those really go out of the bone marrow and they do their final maturation in the thymus. So they take a lot longer to mature. It isn't just done in one little couple-week assay. It needs a lot more changing of media formulations and substrates that those cells see. So lymphoid don't really work the same way. There are different ways to expand those out. So the CFU is kind of one that we recognize when it comes to the myelopoietic pathway that we could really identify in a really short period of time, or at least time that we didn't have to change media. Because if you're doing it in something like methicellulose, or in the old days, they used to do this in sloppy agar for clonal identification, you really can't change the media. So that's sort of the background. For humans, it takes 14 days, and then you get images like you saw on the slide deck. And then you have to score those. You have to go colony by colony. So on an inverted microscope, of course, you need training so that you kind of get used to doing that. It usually takes a couple of months. And it also takes about 5 to 7 minutes a plate in a well, where something like automated can be done in a minute. Anyway, that was a long answer to a short question.
Thank you, Dr. Damen. And does the FDA require the CFU assay?
The FDA, when it comes to guidances, always, and you've seen some of them in this podcast or in this webinar, sorry, they always refer to the CFU like the CFU assay, because historically it was, as you saw, it was defined in 1979. So there's just tons and tons of research to show the applicability of that assay to really quantify those cells. The reality, though, is it is a hard assay to set up, standardize, and then there's subjectivity when it comes to colony counting. In the old days, that was a lot worse because you didn't have necessarily very standardized media products. That has improved, obviously, over the years, so it becomes something that could be standardized. But there's the other criteria around that. How are you counting your cells? Some people absolutely will not use trypan blue. It doesn't give you a good enough viability measurement, for example. So 7-AAD has been suggested as a way to standardize this. So standardizing has been difficult, but as a comparative assay. So if you know your cells up front and you do some manipulation, or even for diseases, patients will have the same CFU output in their bone marrow mobilized peripheral blood until they have a disease. So it's become a very important assay when it comes to that, although the only place that the FDA has actually written it into a guidance, believe it or not, is the CFU assay for the qualification of therapeutic EPO when it's produced. So that's the only place that actually says you must perform a CFU assay. Even for something like PV, it used to be done quite regularly. Now, of course, with PCR and the JAK mutation, there are other ways to identify PV. It can be used for PV diagnosis, but it isn't necessarily required anymore. But so the FDA suggests it a lot, and if you're going to have a good potency assay, they really want you to know that you've done the CFU and that it actually correlates with the CFU because there's so much literature on that. But finding another potency assay has been difficult, and there's been a lot of people looking at that in the last 10, 15 years, and nothing has really stuck as of yet.
Thank you, Dr. Damen. And we have time for just a few more questions, but I want to remind our audience that those questions we are unable to answer live today, and additionally those that come in during the on-demand period, they will be addressed by Dr. Damen via the email address you provided at the time of registration. Okay, let's squeeze in a few more. If the viability and functionality are very different, what is the testing needed for guaranteeing efficacy of the treatment?
Yeah, very good question. It is quite correlative with a fresh product, especially a fresh product that's used within 6 hours. Then viability and potency are very similar, and functional viability, or defined as functional viability. The reality is when you thaw cells especially, or when you leave them too long, everything is dying. If you leave them too long, especially at a really high concentration, the data that I showed here from one of the publications was 48 hours. We don't actually allow anybody to send us samples to test unless they're within the 24-hour window, so recognizing that. But to really test for functionality and not just viability, viability is always just a DNA stain. You have to incorporate some kind of activity, biological activity that takes place in those cells after you've thawed them or after you've manipulated them. Some people, for example, are starting to investigate and have some very good correlative evidence that if you could thaw CD34 cells and stimulate them with IL-3, you can see STAT phosphorylation. I'm a big fan of this. I did a lot of testing on STAT5 to show how important it is to see that you get that activation and phosphorylation so that you really know that you've got this biological function, and it correlates very well with those ability of cells to grow. It's just how do you do that in a standardized way? So to me, it was easier to standardize the CFU, which is why I think a lot of research has focused on that. The reality of having another standardized assay, it would be great to have something that was shorter. Again, always having to compare it to the CFU, but hopefully one day that you can actually see viability, but you have to show some kind of biological function. For T cells, for example, you're going to have to show some kind of effector function, that it actually affects the cells that you say it's going to affect, and not just say, oh, they're viable.
Thank you, Dr. Damen. And we'll wrap with this final question. Why would cells identified as CD34 positive not engraft?
Another good one. Yeah, this has become a lot more recognized recently. So despite, again, the fact, and I was involved in studies in the 90s, so I actually watched it happen live, but more recently, there's still new information that's really allowing us to understand those cells. Sorry, what was the question again?
Yes. Why would cells identified as CD34 positive not engraft?
Right. So CD34 we really recognize, even though originally we thought it was a pure population. Nothing is a pure population when it comes to hematopoietic differentiation. It actually seems more now based on techniques that researchers have been able to employ with single cells and single cell imaging and genetic analysis, that it's a continuum, that the cells start as hematopoietic stem cells, and although on their surface they're still CD34, that's a mixed cell population, and some of the cells are actually already recognizing and becoming fate determined that we can't tell by expression on their cell surface. So there's a lot of things that are going on, epigenetic changes that are happening that we just don't see on the cell surface, that start to split those cells into an engraftable self-renewing population, which is very slow growing because normally a hematopoietic stem cell stays stem by quiescence. So that population is slow growing as it is, and it gets overtaken really by a population that does an asymmetric division, not a symmetric self-renewing division, but an asymmetric division, still CD34 positive, but now is fate committed to one of the lineages and starts to roll down that pathway. So CD34 is sort of encompasses, and it's one of the reasons we changed the definition of hematopoietic stem and hematopoietic progenitors to hematopoietic stem and progenitors, as we've realized over time that both these populations are still CD34 positive, and other markers. It's been difficult, although more recently you've seen them, and I outline them in these slides, that you can start to pick that apart a little bit better and really identify which ones are still engraftable and immature and only doing self-renewing divisions versus those that have done that asymmetric division and are starting to become fate committed.
Thank you, Dr. Damen. Now, before we go, I do want to thank our audience for joining us today and for their interesting questions. There were so many questions we were unable to answer, and again, those will be addressed via the email address you provided at the time of registration. I would once again like to thank Dr. Jackie Damen for her time today and for her important research. We'd also like to thank LabRoots and our sponsor, STEMCELL Technologies, for underwriting today's educational webcast. This webcast can be viewed on demand, and LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. That's all for now. We thank you for joining us and hope you have a great day.
Thank you.